So we're a microfluidics lab. So what is microfluidics? Microfluidics is engineering and science of manipulating fluids on the microscale. We're talking about a scale of a single strand of hair. And so we can build systems, elegant systems, simple or complex multi-layered systems to manipulate cell suspensions. When we shrink the channel dimensions even further down to the size of individual cells, we discovered that these cells start to behave in different ways. So the physics of how these cells behave in the channels becomes very, very interesting. We discovered that we can manipulate or use the inertia of the fluid, viscoelasticity of the carrier fluid to position these cells based on their size, based on their deformability, their softness, based on their shape, and we can use these techniques for separation and isolation of rare cells. We apply these techniques to hematology, where we are isolating particular types of cells from blood, or in oncology, where we're isolating rare cancer-related cells. So the two examples I'll share very quickly is in cancer space. So in liquid biology, liquid biopsy, we use microfluidic devices to isolate circulating tumor cells from patient blood. These are the cells that are shed into circulation by primary tumor, and they represent the genotype and phenotype of the primary tumor. So this approach allows us to sample the tumor without invasive biopsy procedures. And so we use the microfluidic system to trap these cells, rare cells, on the chip, and then use immunofluorescence to interrogate their phenotype or their gene expression. We can also collect these cells off chip and use single cell analysis for further characterization. In another microfluidic example, we are developing systems for building models of cancer. So this is the chips that allow us to use 3D culture methods to culture patient-specific organoids, patient-specific tissues, and use them for drug screening. In the example that you see there, we can create large arrays that that are microfluidically addressable, and we can deliver specific drugs and treatments to individual organoids and look at their response. And the example that you see there, I can't quite point to the ones, but the example that you see there is two types of tissues, the F231 and F671. These are the two patient-specific organoids that have the same underlying mutation they carry as G12C. But however, as you can see, they respond differently to treatment. And that's as it's shown by the green core of one of these, which means that these are more resistant to treatment than in another patient. And so we can use these systems to study drug resistance and patient-specific response. And finally, let me highlight the National Science Foundation Center that I co-direct, and this is a joint venture between UIC and UCI. This is a perfect arrangement of letters. And we just celebrated a 10-year anniversary, and we've been very successful in securing follow-on funding. We've had multiple patents and technologies developed in the center, and very importantly, we've trained a large number of students, undergraduate students in microfluidic technologies. Thank you.